So in lesson 4, we studied the major problems encountered in the cell culture lab, that is contamination. Other than that, various minor issues with cells occur and frustrate the researcher. Sometimes cells grow very slowly, or sometimes their morphology changes, and there are numerous reasons for that. One of them being cross contamination with other cell lines, and sometimes there are no viable cells after we thaw the cryopreserved cells, although we had preserved them well before. The reasons for these issues are that uh, sometimes cells are not stored correctly, or they are not thawed correctly. Sometimes the thawing media is not properly made, or the growth media is not correct, or sometimes the FBS, the serum we are using, is of poor quality, or the antibiotics are not working, due to which the contamination occurs, bacterial contamination mainly. And sometimes the cells have been passaged too many times, or the cells were allowed to grow beyond their confluence state. And these were the most common cells related issues. So what are the most common lab related issues? The issues that researchers face in the lab other than cells are the contamination of consumables, for example media contamination, FBS contamination, PBS contamination, and sometimes pellet, the pellet which is formed after centrifugation, the pellet of the cells, it gets detached upon pipetting, the supernatant, as we have discussed in the previous lesson, the protocols 1. And there are problems during the process of cell counting, when during the process of cell counting when we have to plate them for passaging or for assays, etc.